Good morning, everyone. My name is Nataliya Razumilava. And first of all, I want to thank organizers for giving me an opportunity to present at this outstanding meeting. I am a physician scientist at University of Michigan, and I see patients with... Good morning, everyone. My name is Nataliya Razumilava. And first of all, I want to thank organizers for giving me an opportunity to present at this outstanding meeting. I am physician scientist from University of Michigan, and I see patients with cholangiopathies and liver cancer in the clinic. In my lab, I study mechanisms underlying biliary regeneration and cancer. Today, I will talk about organoid models of cholangiocarcinogenesis with focus on biliary progenitor cell role in cholangiocarcinoma, organoids as a model of biliary progenitor cells, and share with you our project on Wnt signaling regulation of biliary progenitor cells. As we know well, biology of cholangiocarcinoma is not well understood. Thus, we don't know what changes epithelium undergoes when it transitions from normal, shown on the left, to cancer, shown on the far right. But we do know that there are conditions that are risk factors for cancer development. These are called cholangiopathies, and as you heard earlier today, this includes primary sclerosing cholangitis. It also includes choledochal cysts. What we also know that during epithelium transition to cancer, peribiliary glands, which are home for biliary progenitor cells, undergo hyperplasia, and peribiliary gland hyperplasia is associated with biliary dysplasia, which is last step before full-blown cancer development. If we know what causes peribiliary glands hyperplasia, can regulate it, we will have more opportunities for early diagnosis, chemoprophylaxis, and treatment for cholangiocarcinoma. Thus, in my lab, our main question is, what are the mechanisms regulating biliary progenitor cells hyperplasia? We use following approach: acute biliary injury model in mice, human and mouse organoids. We manipulate Wnt signaling pathway with pharmacological inhibition, such as IWR1 endo. Wnt signaling and regulation of liver progenitor cells was suggested based on liver studies. And we turn our focus on Wnt signaling as its highly conserved pathways that regulates tissue growth and development. It regulates cell proliferation and progenitor cell function based on intrahepatic bile ducts and liver studies. It's quite complex pathway. It includes 21 Wnt ligands, respondin, which amplifies Wnt signaling, and beta-catenin, which is transcriptional effector of Wnt signaling pathway. Wnt targets are CD44 and SOX9. Our analysis of TCGA patient database showed that at least in intrahepatic cholangiocarcinoma, there is upregulation of Wnt ligands 7A and 7B, shown on the left, and on the right, there is upregulation of Wnt signaling targets such as CD44 and SOX9. In our mouse models of acute biliary injury, when we ligate mouse bile duct close to duodenum to induce its injury, we analyzed extrahepatic bile ducts for cell proliferation and observed that there is increase in proliferating cells, shown in the green and marked by Ki67. It was also associated with increase in Wnt ligands, shown on the right bar diagram, after bile duct ligation. But is it relevant to progenitor cell function? Yes, it is. We analyzed bile ducts once again in our animals after bile duct injury and examined it for PDX1 progenitor cell marker. It was expressed in peribiliary glands at baseline, shown on the left, and after bile duct ligation, it was significantly increased in peribiliary glands as well as in epithelial cells. We quantified our findings on the right and also confirmed findings of increased PDX1 on messenger RNA level, shown on bar diagram on the right. This is in vivo studies, which can be cumbersome and expensive, but can we model biliary progenitor cells in vitro? Organoid represent just perfect model, as per definition it is three-dimensional structures derived from stem and progenitor cells. They were first generated from gut and later on from hepatocytes. They were also generated from intrahepatic and extrahepatic cholangiocytes. They can be established from different sources, such as surgical resection samples, biopsy, autopsy, endoscopically derived samples, and frozen tissues from biobanks. They were already tested for translational applications such as regeneration, gene editing for disease correction, as a drug testing platform, and as carcinoids, which are organoids from liver cancer, which can be tested again on a plate or in vivo in cancer avatars. In my lab, we work mainly with endoscopically derived samples, bile, biliary brushings biopsy, which can be readily obtained during ERCP. On the upper panel, you can see bile samples, cytological brush samples, and biopsy samples. We collect cells and wash them, embed in matrigel, and plate in the presence of growth factors, which support progenitor cell function, such as Wnt, respondin, gastrin, HGF, EGF, and FGF10. Lower panel shows representative images of organoids derived either from bile, brushings, or biopsy. They look all about the same. As access to human tissue is limited, and my lab specifically focuses on extrahepatic bile ducts, we also generated organoid models from mouse extrahepatic bile ducts. Last year, we reported protocol and showed that extrahepatic bile duct mouse organoids express markers of progenitor cells such as PDX1, shown on the upper panel, SOX9, shown on the lower panel in immunofluorescence images, as well as on messenger RNA level on the right. They also express markers of more mature biliary cells such as CK19, CFTR, aquaporin, ecadherin. Importantly, they express cilia marked by acylated alpha-tubulin, signifying cell polarization and directionality of transport, and meant that organoids have lumen. Organoids are often cultured in the presence of Wnt in most epithelial systems, but isn't required for mouse extrahepatic organoid growth as a readout for progenitor cell function. To test it, we put organoids through serial passaging and cultured them in the presence or absence of Wnt. As you can see, organoids that were plated with Wnt had normal plating efficiency, shown in white bars on these bar diagrams, on the left and the right, and normal growth, while organoids plated without Wnt failed to establish as we kept passaging them and stopped growing, shown on the gray bars. While prior experiments showed that organoids require exogenous Wnt for growth and progenitor cell function, what is the source of Wnt signaling in extrahepatic bile duct? We generated specifically fibroblast culture from extrahepatic bile ducts in mice and cultured cholangiocytes and fibroblasts together. Our readouts were cell proliferation and organoid growth. Organoids cultured with fibroblasts had significant increase in growth, shown on the right, as well as cell proliferation. We quantified proliferating cells by counting EDU positive cells in our organoids. But do fibroblasts secrete these factors supporting organoid growth? And do they really signal via wind? We used following approach. We cultured fibroblasts and cholangiocytes separately, collected medium from fibroblasts, and placed it on cholangiocytes. We also blocked Wnt signaling by ILWR1 endo, which prevents beta-catenin translocation to nuclei. As you can see, organoids that were cultured in fibroblast-conditioned medium had significantly increased growth on a picture on the left lower corner, and diagram blue, third bar from the left. When we added beta-catenin translocation inhibitor, organoids expressed decreased growth. In summary, acute biliary injury is associated with increased Wnt signaling and progenitor cell function. At least in mice, mouse fibroblasts secrete factors that stimulate extrahepatic bile duct organoid growth, as well as proliferation. And these processes are Wnt dependent and require cell-to-cell interaction. This is mouse studies, but is it relevant to human diseases? To answer these questions, we use organoids derived from patients with benign and neoplastic cholangiopathies. Here we show you our experiment when we measured organoid plating efficiency in organoids derived from patients with PSC and non-PSC. PSC-derived organoids had increased progenitor cell function, evidenced by increased plating efficiency. In our mouse studies, we learned that biliary progenitor cell function requires Wnt, but does it require for human biliary organoid? As you can see, non-PSC and PSC organoids that were cultured without Wnt showed decreased plating efficiency and, importantly, failed to grow. This set of experiments allowed us to conclude that biliary organoids can be generated from different sources and can model biliary progenitor cells in health and disease. They function through Wnt-dependent pathways and require cell-to-cell interaction. Our future directions are further dissecting cell-cell interaction using power of genetic mouse models, expand our human biliary organoid biobank, and generate co-culture models with immune cells. I want to acknowledge my funding agencies, NIH, Gilead, ASLD. Importantly, I want to thank patients who generously donated their samples and worked with us. I want to thank my mentors, Dr. Linda Samuelson, Merchant, Drs. Allen and Spence from University of Michigan, my outstanding clinical support, Dr. Sturgeon and Wamsteker, my collaborator, Dr. Wells from UPenn, and my team members, Noreen Mahammadzaki and Junya Shiota. Thank you.
Firstly, thank you for inviting me to speak to you all today, and thank you very much to the organizers for taking the time and effort to put all of this online. It's a massive undertaking, and I'm delighted to be able to speak to you all from Edinburgh today and talk about some of the work that we've been doing over the last couple of years. So the title of my talk really is to investigate patient-led mutations in genetics and try and understand how those genetics contribute to tumor formation and tumor growth. So my lab actually works on how ducts form and grow both in the embryo but also in the adult following injury. And we're really interested in the signaling mechanisms and processes that allow that to happen and allow that to kind of repeat itself, I guess, throughout life. But more lately, we've been really looking at how those repair mechanisms and how those developmental mechanisms interact with oncogenic mutations in cancer. And what we realized quite early on is that for cholangiocarcinoma particularly, the information we had about how these tumors grow and what driver mutations there are that promote tumor initiation and growth was pretty lacking. So this is the work of Nicholas Younger when he was in the lab, and Molly Wilson, who's taken up the mantle. And really what they've been doing is investigating how we can integrate the mutational information that we have from whole exome and whole genome sequencing studies into animal models. We did, of course, know that there were a handful of mutations that have been previously described through numerous studies. So highlighting just some of the older ones here, actually, from fluke-associated cholangiocarcinoma and spontaneous cholangiocarcinoma, we had these ideas that there were the mutations such as IDH, RNF43, P53, and KRAS were recurrent within these tumor populations. However, it wasn't clear to us whether there was a high level of genetic heterogeneity that also occurred within these tumors. The other concern we had during this, I suppose, the early days of the study was that actually the data that we had was pretty poor. The exome sequencing studies had a very low sample size. Tumors from both fluke-associated and spontaneous cholangiocarcinomas were included in the sequencing data, and also extrahepatic and intrahepatic cancer was both included. And so actually that gave us a very kind of heterogeneous pool of data to try and understand the genetic components of these cancers. And also, because of the low level of, or the low numbers of tumors, frequency-based approaches to identify mutations really have fallen short. Yes, we identify these very recurrent things like P53 and RAS and IDH, but we struggle to identify many of the other oncogenes that might be playing a role in tumor initiation and growth. There are some incredible efforts to target therapies to particular mutations in cholangiocarcinoma, particularly the FGFR fusions, IDH1 and 2, and ARID1A. And those have been studied, but many of the drug development regimens and I suppose the drug development pipelines for cholangiocarcinoma are driven by these genetic rearrangements in other tumors like lung adenocarcinomas and glioblastoma. So we really don't know what the kind of genetic landscape is functionally in cholangiocarcinoma, and whether actually we can manipulate this therapeutically in order to give a kind of a therapeutic benefit. What many cholangiocarcinoma exome and whole genome sequencing studies have identified is that cholangiocarcinoma is hugely heterogeneous. So whilst we are comfortable with the idea that there is a canonical group of driver mutations that are recurrent in about a quarter to a third of patients, many of the patients that we see don't have these, or indeed have these in addition to a number of other mutations. So this is the problem, because we don't know whether the presence of a targetable alteration predicts patient response. So just because you have an FGFR translocation, does that mean you are responsive to an FGFR inhibitor? Actually, not always. And that might be because you've got interactions between commonly mutated and rarely mutated genes, which modify the tumor phenotype. And whilst we do have this canonical group, about a third of patients don't have a mutation in any of those drivers. And this leads us to the idea that, well, maybe there is a group, or there are groups of weak drivers within these infrequently mutated genes that act together to promote tumorigenesis, initiate cancer growth in the absence of a strong driver. And so this is really what we sought to kind of study and set out to look at. Does this long tail of infrequently mutated genes contain driving mutations that act together to induce tumorigenesis? And actually, I'm not going to have time to discuss that today. But what I will look at is whether these mutations interact with more common drivers to modify the tumor phenotype. To do this, we took a bit of a step back. So as I'd already mentioned, there are a number of exome and whole genome data sets out there in the literature. And we thought that what we would do is try and rationalize and reaggregate these data sets so that we can really understand the genetic complexity of intrahepatic cholangiocarcinoma. To do that, we took whole genome and whole exomes from intrahepatic cholangiocarcinoma patients only. We identified the data sets, we aggregated those, and where we could, we realigned reads against the human genome and recalled any variants to try and give us a really high confidence data set that we could then take forward. As I've mentioned already, there are problems using frequency dependent measures for looking at, I suppose, recurrent mutations in small numbers of patients or relatively small numbers of patients. So we utilize this frequency independent driver gene prediction tool called Integen. And Integen works really in three ways. It identifies whether a mutation at a particular site is recurrent, so is likely to be a gain of function mutation. It looks at whether mutations fall into particular regulatory sites, such as catalytic domains or promoter regions of a gene. And it also addresses whether a gene has a mutation rate higher than the background mutation rate of the DNA within that cancer. And so from that, we've been able to identify a list of potential oncogenic mutations that occur within patient cholangiocarcinomas. And unsurprisingly, what we found was the canonical data set, or reassuringly, what we found was the canonical data set that has been previously described. So that's IDH1, ARID is there, BAP, P53, KRAS, IDH2, and PBRM1. So we were very happy that our tool could find what has been previously found. We also identified a fairly large number of mutations that were available in cosmic, so that is to say that they have been identified as potentially oncogenic in other cancers, not only cholangiocarcinoma. And within this, we find things like SMADs, NRAS, NCOR. And then we found another group of mutations which we've identified as novel. So these are things that haven't been previously identified in cholangiocarcinoma as potential tumor oncogenes, but have also not been found, or at least not been listed in the cosmic database. And within all of these different groups, there are about 6 regulatory families that kind of come out with our pathway analysis. And this is important because it suggests to us that these novel or rare mutations are able to interact with proteins that are mutated more commonly in order to affect their phenotype and potentiate their biology. So my lab is definitely not a bioinformatics lab. We are a wet lab where we use animal models to try and understand disease processes. So having identified about 96 genes that are mutated from just under 300 patients, we then sought to model whether we can incorporate some of that genetic heterogeneity into our mouse cancers. So to do this, we adapted a system known as a hydrodynamic tail vein injection, where we can deliver plasmids under high pressure through the mouse tail vein that then enter into the liver, and in the presence of transposase, we can edit genes or we can overexpress oncogenic cDNAs. And what we did here is we built a guide library of guides that would interfere with the patient mutations, or the genes that we found mutated in patients. And we did this alongside KRAS and NRAS. And the question we really wanted to address was which of these mutations synergize with NRAS or indeed KRAS to promote tumorigenesis. So we didn't know initially whether we could edit the endogenous genes within a mouse in vivo. So we've established this Cas9 CRISPR gene editing system where we're able to express a guide RNA that targets LOXP. So this is the sequence of DNA that flanks the tomato gene in a reporter mouse and deletes it, allowing read-through of EGFP. So essentially we can lineage trace genetic edits. And you can see here in panel D that when we administer both the Cas9 and our guides against LOXP, that we can edit liver cells specifically. So this gave us confidence that actually when we put in our guide library, which contains guide RNAs against genes that are lost in patient cholangiocarcinomas, that we would be able to do this in vivo. So what did we find? What we were able to show was that if we expressed NRAS G12V mutants or KRAS G12D mutants on their own, so that's the first two livers, the first and the third liver in the whole liver pictures, or the dots on the left-hand side of both of those graphs, that we didn't see any tumors at all. And that's not surprising given that we know RAS mutant cells are cleared by macrophages. And that's been shown previously by a number of groups. What was interesting, we thought, is when we co-administered our library of guide RNAs with either the KRAS mutant or the NRAS mutant, we were able to initiate cancer in those livers. And those cancers within 10 weeks grew into macroscopic tumors, as you can see, essentially in liver 2 and 4. And those tumors were very sizable, so they rapidly overwhelmed the hepatic function of the mouse. Those cancers are lineage traced, so they also express a GFP, and you can see in the bottom panels that our tumors are GFP positive and that the tumor cells are distributed amongst a very dense immune and fibrotic stroma. Very much reflecting, we think, what we see in patients as far as the physiology of the cancer goes. But to identify what guide RNAs were causing this deletion and these alterations, we moved to sequence these cancers with exome sequencing and identified in yellow a number of genes that were lost in the presence of KRAS-driven cancer, and in blue, a number that were lost in the presence of NRAS-driven cancer. We have specifically focused in this middle histogram where we found genes that were recurrently lost in both the NRAS and KRAS groups. And unsurprisingly, our top hit is P53. We kind of expected it would be, but neatly, we have some exciting candidates that have come out of this screen. So BRCA, NF2, plexin, a number of things that actually might alter the tumor phenotype and also tumor progression. And we have very particularly focused on NF2. So this is neurofibromatosis 2. It's previously been described in mouse at least as promoting tumorigenesis if it's deleted in the embryonic liver. But we're now interested in whether this is an oncogene that interacts with RAS in order to promote tumor progression de novo in the adult. And Merlin is historically, I suppose, or classically known as a negative regulator of the Hippo pathway. So deletion of Merlin or NF2 causes hyperactivation of Hippo, but recent data shows that actually it's involved in many other processes. So it regulates the Lin 28B LET7 pathway. It's known to interact with mTOR and also a number of cadherins at the tight junctions and the adherent junctions between cells. So we're interested now in whether dysregulation of NF2 or all NF2 associated pathways are oncogenic in the presence of RAS. And when we delete NF2 on its own, we do see an additive phenotype. So what here we have on the left is a mutant KRAS with p53 loss caused by Cas9 CRISPR, and we can see large numbers of focal discrete tumors. However, when we add a deletion of NF2 into that, the tumors become much more invasive, they're much more highly migratory, they invade most of the liver within 14 days, and ultimately kill the mouse very quickly. So what we are hoping for is that by using these very extreme tools and these very extreme contexts to look at tumor development and tumor initiation, that we might find something that is more broadly applicable to cholangiocarcinoma progression and cholangiocarcinoma growth. Finally, we have started to look at what changes are downstream of NF2 loss in these cancers. And interestingly, and perhaps surprisingly, we've found a downregulation of a number of cadherins and protocadherins. And this is something we are now looking into further. So we know that protocadherins and non-classical cadherins are involved in cell polarity and migration, but these are classically cadherins that are found in the neuronal system, but actually they might have a kind of a role in cancer and particularly migration of cancer cells. So this is something that we're hoping to investigate further. With that, I would like to thank again the Cholangiocarcinoma Foundation for inviting me to speak today. Give credit to my lab, particularly Molly Wilson, who's my current PhD student, and Nick Younger, who left earlier this year, having completed his PhD, for their hard work in integrating the patient data into our animal models. And we hope that next year we'll be able to come along to the meeting and give you a more complete story of what's come out of this final step.